Welcome to Alacrity Explains Medicine. Today we are going to be looking at cell death, where we'll be looking majorly at apoptosis and necrosis. So, cell death is a fundamental endpoint of cellular injury. So, when a cell undergoes stress, when there's a stressor on a cell, that cell tries to adapt. In the course of adaptation, if the stressor is not removed, or the cell fails to adapt, or exceeds its ability to adapt, the cell gets injured. So this injury can either be reversible injury or an irreversible injury. If the injury is irreversible, the cell progresses into what? Cell death, where the cell dies. So cell death is the end point of cell injury, specifically irreversible cell injury. It occurs through two distinct mechanisms, which is apoptosis and necrosis. Apoptosis is a programmed, energy-dependent type of cell death, while necrosis is accidental, energy independent, and always pathologic. Necrosis is always pathologic. So while both result in cell death, they differ entirely in their etiology, morphology, biochemical pathways, and host response. So if you look at this image, it's a normal cell which has undergone stress, has adapted, and in the course of adaptation, this cell has been injured, and this injury is irreversible. So the cell will either undergo apoptosis or undergo necrosis. By necrosis, to follow this pathway and lead to destruction of the cell. By apoptosis, same thing, follow a different pathway and lead to destruction of the cell. So now we are going to look at apoptosis. To look at apoptosis, then we look at necrosis. Now, apoptosis is a highly regulated, active form of cell death that eliminates unwanted, damaged, or infected cells without eliciting an inflammatory response. There is no inflammatory response in apoptosis. It is essential for the normal development and tissue homeostasis. It's essential for normal development as well as tissue homeostasis. It could be physiologic or pathologic, unlike in necrosis, where we said necrosis is always pathologic. For physiologic, we have programmed destruction during embryogenesis, that is removal of webbing between the digits during embryogenesis. We have hormone-dependent involution, for example, in endometrial shedding during the reproductive life of a lady. We also have pathologic apoptosis. In pathologic apoptosis, for example, we have DNA damage by radiation, chemotherapy, or toxins. Typically, the p53 is mediated by the p53 gene. So protein misfolded proteins are being removed by apoptosis. We also have viral infections like hepatitis, cell death in certain tumors. There is response due to response to therapy. So apoptosis in these situations are said to be pathologic. Now, what are the morphologic features of apoptosis, the hallmarks? What do you see in a cell that is undergoing apoptosis? First, we have what? Cell shrinkage. So the cell volume, the volume of the cell decreases. A cell undergoing apoptosis, its volume decreases, right? So cytoplasm becomes dense and eosinophilic. We also have chromatin condensation, where the nuclear chromatin aggregates peripherally around the nuclear membrane. There's aggregation of the nuclear chromatin around the nuclear membrane, forming crescentic or dense spherical masses, which we call this pycnosis. It is followed by a nuclear fragmentation called karyohexis. Pycnosis followed by karyohexis. Karyohexis is nuclear fragmentation, while pycnosis is a situation whereby the nuclear chromatin aggregates peripherally under the nuclear membrane. We also have apoptotic bodies. Apoptotic bodies are seen in apoptosis. These are cell fragments. The cell will fragment into membrane-bound, intact apoptotic bodies containing a cytoplasm and a nuclear fragment. Note, the apoptotic body contains an intact cytoplasm and a nuclear fragment, so the membrane is not disrupted. Then phagocytosis occurs. These apoptotic bodies are now rapidly engulfed by adjacent cells or macrophages with no surrounding inflammation. Remember, we said there's no inflammation in apoptosis. Intact cell membranes prevent the leakage of cellular content. The apoptotic bodies, we see the cell membrane is what? Intact. So this prevents leakage of cellular content into the surrounding space. So this is an image showing us how apoptosis occurs. We have a normal cell here which progresses to cell shrinkage and nuclear condensation. From there we are moving to what? Cytoplasmic blebbing. There are presence of blebs. These are blebs on the surface of the cell. See blebs. Then there's packaging of cell organelles in these blebs. Cell organelles, after fragmentation and condensation, they are being packaged in these blebs that are formed on the surface of the cell. Then these blebs now separate to form what? Apoptotic bodies. These apoptotic bodies are what we were describing earlier. Then phagocytosis occurs by macrophages. They phagocytose these apoptotic bodies. Now, what's the biochemical pathway or mechanism of apoptosis? The mechanism of apoptosis follows the caspase cascade, a cascade formed by caspases. Now, it is execution. The execution of this apoptosis is mediated by a family of 16 aspartic proteases, which we call them caspases. These caspases are a family of 16 aspartic proteases which mediate or execute apoptosis. We have the initiator caspases and we have the executioner caspases. The initiator caspases include caspase 8, caspase 9. While the executioner caspases, we have caspase 3, caspase 6, caspase 7. Now, these initiator caspases, what they do is they activate the executioner caspases. The executioner caspases now lead to what? Cleavage of structural proteins that will activate endonucleases. This will cause degradation of the cell. So now let's look at the two major pathways of apoptosis. We have the extrinsic pathway and the intrinsic pathway. The extrinsic pathway is also called the death receptor pathway, while the intrinsic pathway is also called the mitochondrial pathway. Now, for the extrinsic pathway, what happens is that you need ligands. Ligands will bind to what? Death receptors. These death receptors are on the surface of the cell. They're on the cell membrane, the surface of the cell. So, by binding of these ligands, we have the fast ligand, we have the TNF, which is the tumor necrosis factor. They bind to death receptors, the fast receptor or the tumor necrosis factor receptor 1. When these ligands bind to the receptor, look at this image. From this image, we're talking about the extrinsic pathway, which you can see here, the death receptor pathway. So receptor ligand interactions, either the fast ligand or the tumor necrosis factor receptors, they are on the surface of the cell. On the surface of the cell. So ligands will bind, this is a ligand, will bind to these receptors. When they bind to these receptors, there's an adapter protein, which will now what? Stimulate the formation of what? Initiator caspases. Now, this is what we're talking about. The ligands bind to the receptors, recruiting what? An adapter protein, which will now activate caspase 8. Remember, we say caspase 8 is an example of what? It's an example of an initiator caspase 8. Now, that's the extrinsic pathway. For the intrinsic pathway, which is the mitochondrial pathway, it is triggered by DNA damage, hypoxia, or stress. Look at this image. You have growth factor withdrawal, when there's withdrawal of growth factors, DNA damage by radiation, toxin, free radicals, protein misfolding. All these are what? Triggers that will trigger the mitochondrial pathway of apoptosis. This is the mitochondrial pathway of apoptosis. So what happens is that it results in the mitochondrial outer membrane permeabilization. The mitochondrial outer membrane becomes permeable. When it becomes permeable, it releases cytochrome C into the cytosol. There's release of cytochrome C into the cytosol due to the increased permeability of the outer mitochondrial membrane. So cytochrome C is released into the cytosol. When cytochrome C gets to the cytosol, it now forms what? An apoptosome. How does it form this apoptosome? It binds to APAF1. APAF1 stands for apoptosis activation factor. Apoptosis activation factor 1. So cytochrome C binds to APAF1 to form what? An apoptosome. Then there's now formation of an apoptosome. Apoptosome now activates caspase 9. Remember, we say caspase 9 is also what? An initiator caspase. So in the extrinsic pathway, caspase 8 is the initiator caspase. In the intrinsic pathway, caspase 9 is the initiator caspase. When this initiator caspase is now formed, we now have the executioner caspases, which is caspase 3, 6, and 7. So the initiator caspases will now activate the executioner caspases through a common pathway. Now, this intrinsic pathway or mitochondrial pathway is tightly regulated by the BCL2 family proteins. In the BCL2 family proteins, we have pro-apoptotic proteins, that is the BACs and the BAC. We also have anti-apoptotic, the BCL2, the BCL-XL, and the MCL1. So what these proteins do is that the pro-apoptotic, they promote apoptosis, while the anti-apoptotic inhibit apoptosis. So there has to be a balance. There is always a balance between the pro-apoptotic and the anti-apoptotic factors. If there is an increase in the pro-apoptotic factors and a decrease in the pro-apoptotic factors, there is now what? Stimulation of what? Apoptosis. Apoptosis is now stimulated. So if you look at this pathway, we have the BCL2 family proteins, the BACs and BAC. Remember, with BACs and BAC, we said they are what? They are pro-apoptotic. When we have the regulators, which are anti-apoptotic, the BCL2, the BCL-XL family. So what they do is that they regulate, they release this pro-apoptotic are stimulating the mitochondria to release cytochrome C. You get the BCL2 family, the effectors of apoptosis, the BACs and the BAC, they stimulate the mitochondria to release cytochrome C. While the anti-apoptotic, which is BCL2, BCL-XL, what they do is that they inhibit the formation of what? Pro-apoptotic proteins. But in the case of this stimulus, there's release of cytochrome C. Cytochrome C, remember we said what? Form the initiator, activate the initiator caspase, caspase 9, through the formation of an apoptosome by binding to APAF1. So there is now initiator caspase, which will now produce the executioner caspase. The pro-apoptotic proteins also promote the formation of executioner caspase. This executioner caspase will now activate what? Endonucleases, as well as breakdown of cytoskeleton. This cytoskeleton, breakdown of cytoskeleton, as well as endonucleases, which cause DNA fragmentation, will promote the formation of cytoplasmic blebs. These cytoplasmic blebs will now form what? Apoptotic bodies. These apoptotic bodies are now engulfed by phagocytes through the process of what? Phagocytosis. So this is both the mitochondria or intrinsic pathway and the extrinsic or death receptor pathway of apoptosis. Now they are moving to necrosis. Necrosis is an uncontrolled passive form of cell death resulting from what? A severe irreversible injury. There's a severe irreversible injury. It is always pathologic, and it is characterized by what? Cell swelling, membrane rupture. Remember, in apoptosis there is no rupture of membrane, but in necrosis there is membrane rupture and prominent inflammatory response, which we don't have in apoptosis. What are the causes of necrosis? We have severe hypoxia or ischemia, extreme physical or thermal trauma, chemical toxins, infections, and complement mediated injury. So we have morphologic features of necrosis, and these morphologic features include cell swelling, that's loss of cell membrane integrity, leads to an influx of what? Water and ions, causing cytoplasmic swelling. The swelling of the cell, which we would refer to as cellular edema. After the cell swells, the cell membrane loses its integrity. There's loss of membrane integrity. The plasma membrane and organellar membranes become what? Disrupted, which means the membrane of the organelles as well as the plasma membrane gets disrupted. That's another morphologic feature of necrosis. We also have nuclear changes. There are three sequential patterns. We have pycnosis, which is nuclear shrinkage and increased basophilia, followed by karyohexis, which is fragmentation of the pycnotic nucleus. Then finally karyolysis, which is a complete dissolution of the nucleus due to DNA's activity. Remember, in apoptosis we mentioned the pycnosis and karyohexis, but there's no karyolysis. So another change, another morphologic feature you have is what? Cytoplasmic changes. There's increased eosinophilia. There's also myelin figures, and maybe myelin figures may be visible due to phospholipid aggregates. We have inflammation. There is inflammation in necrosis. There's leakage of intracellular content into the extracellular space, which triggers an intense acute inflammatory response. So this is a normal cell, has a reversible injury. This reversible injury, if the cell recovers, it goes back into the normal cell. If the injury becomes irreversible, don't mind this image, if the injury becomes irreversible, it undergoes what? Necrosis, and there is inflammation in necrosis. So the biochemical mechanism, basically there is a loss of ATP, because you know that the cell membrane contains sodium, potassium, ATPase pumps, which need ATP for their functioning. So when the cell loses ATP, it leads to failure of this sodium, potassium, ATPase pumps. This now causes influx of what? Calcium, as well as activation of phospholipases, proteases, and generation of reactive oxygen species. These phospholipases will degrade phospholipids, proteases, and reactive oxygen species will cause cell injury to the cell, cause more damage to the cell. This culminates in a widespread membrane damage, now resulting in damage to the cell membrane, which in apoptosis we don't have damage of the cell membrane. So morphologic patterns, these ones you must know it. You must know this. You have to memorize it and know it. Morphologic patterns of necrosis. The two major morphologic patterns of necrosis are coagulative necrosis and liquefactive necrosis. So in coagulative necrosis, it is the most common. It is seen in solid organs. The heart, the kidney, the spleen. You can remember this using KSH. K for kidney, S for spleen, H for heart. KSH, solid organs, following what? Ischemia. It occurs following ischemia. The architecture of the organ affected is preserved for days. There is preservation of the architecture. The cells appear eosinophilic with loss of nuclei. It proceeds towards granulation tissue and fibrosis. Proceeds to the formation of granulation tissue and fibrosis. Second is the liquefactive necrosis. Liquefactive necrosis is seen in brain infarct, majorly seen in brain infarct. It can also be seen in bacterial abscesses like amoebiasis and as well as fungal infections. There is enzymatic digestion. This enzymatic digestion completely dissolves the tissue. Enzymes are released. These enzymes now what? Digest the tissue. They digest the tissue, forming liquid cystic cavity filled with pus or debris. Liquid cystic cavities which are filled with pus or debris. So this is an image of coagulative necrosis. You see that the architecture is preserved. This is an area of infarct, and this is on histological slide an area of what? Coagulative necrosis. And in this second image we have what? Liquefactive necrosis. See, this is the brain. See the gyri and the sulci of the brain. So this is the brain. This is liquefactive necrosis. This is a normal brain on histology, and when liquefactive necrosis occur, you see how it appears. Good. So moving to caseous necrosis. So caseous necrosis is characteristic of what? Tuberculosis. Take note. It is characteristic of tuberculosis. The combination of what? Coagulative and liquefactive necrosis. Caseous is a combination of coagulative and liquefactive necrosis. It appears grossly as friable cheese-like white material. It has a cheese-like appearance. The caseous necrosis has cheese-like appearance. Histologically, it shows a cellular eosinophilic granular debris surrounded by what? Granulomatous inflammation. We see the Langerhans giant cells. Take note, it is not Langerhans. Langerhans are seen in skin. There are cells in the skin. It is not Langerhans. It's Langerhans. As you can see, take note of the spelling. It is Langerhans giant cells. Then for fat necrosis, it occurs in adipose tissues, typically acute pancreatitis. We have lipases, that are enzymes, digestive enzymes. They are released. They release fatty acids that binds to calcium and cause saponification, forming a chalky white area. In caseous necrosis, has a cheese-like appearance. In fat necrosis, we have a chalky white area. Histologically, it shows shoddy outlines of necrotic adipocytes with basophilic calcium deposits. This is caseous necrosis, has a cheese-like appearance. You can see on histology, this is what caseous necrosis, while for fat necrosis, you can see that fat cells. You can see how fat it looks, and you can see presence of fat cells on histology. These are fat cells. These are fat cells. These are fat cells. So this is fat necrosis. Lastly, we have fibrinoid necrosis. Fibrinoid necrosis is seen in immune-mediated vascular damage, polyarteritis nodosa, as well as malignant hypertension, seen in the deposition of fibrin in the wall of the blood vessels. So fibrin deposition in the vessel walls appears as bright eosinophilic amorphous material, giving a fibrin-like appearance. It gives a fibrin-like appearance. So this is your deposition of fibrin in the wall of blood vessels. So we have looked at apoptosis, its morphologic features and the mechanism. We have looked at necrosis, its morphologic features and the forms of necrosis, as well as the mechanism of necrosis, the biochemical changes in necrosis. Now, what are the differentiating points between apoptosis and necrosis? Apoptosis is triggered by what? Physiologic signals or mild pathologic stimuli. While for necrosis, it is triggered by what? Severe, irreversible pathologic injury. Energy requirement for apoptosis is active, which means it's energy dependent. It is ATP what? Dependent. It's an active process and it's ATP dependent. Necrosis is a passive process and it is ATP independent because ATP is depleted, so it's not dependent on ATP. Cell size for apoptosis, the apoptotic cells shrink, so there's decrease in the cell size. For necrosis, the cells swell, so there's a volume gain, there's increase in size, the volume. For membrane integrity, in apoptosis, it maintains an intact membrane until fragmentation into apoptotic bodies. There's an intact membrane until fragmentation into what? Apoptotic bodies. For necrosis, there's loss of membrane integrity very early. The membrane integrity is lost very early in the process. The nucleus, apoptosis shows condensation and fragmentation, that is pycnosis and karyohexis, without karyolysis. There is no karyolysis in apoptosis. While for necrosis shows pycnosis, karyohexis, as well as karyolysis in that order, follows that order. So for host response, apoptosis is non-inflammatory. Phagocytosis occurs without inflammation, while for necrosis it is highly inflammatory due to leakage of cellular content. Because of the membrane damage, there's leakage of cellular content, which triggers what? An acute inflammation. For DNA fragmentation, apoptosis produces a characteristic internucleosomal cleavage into fragments, while for necrosis it produces a random smeared DNA fragmentation. Tissue effect apoptosis affects scattered individual cells. Necrosis affects large contiguous sheets or group of cells. Necrosis involves group of cells, while for apoptosis involves scattered individual cells. Pathologic versus physiologic apoptosis occurs in both physiological and pathological settings, while necrosis is always pathological. Thank you for watching. That will be all for this video. Don't forget to like, comment, and subscribe to Alacrity Explains Medicine for more interesting videos. Thank you.